Well, welcome back, everyone. And if you are new to the conference, welcome to the 2021 Annual Conference of Western Field Ornithologists. As you all know, the central purpose of WFO is to promote the study of field biology of birds. And as a result, a feature of our annual conferences has always been a set of science sessions where people give short presentations on current active research. And so we are doing the same thing in the virtual conference and have a series of science sessions arranged over the length of the conference. WFO also in the last couple of years started a grant program to support graduate and undergraduate students in support of their research. And I'm delighted that we'll have two speakers who are recipients of our first 2 years of research grants. So with that, I'll turn it over to Steve Rottenborn. Steve has been a board member of WFO and has been chair of the science sessions for a number of years. And so I'll hand it over to you, Steve.
Thanks a lot, John. Before we get started with the science sessions, I just wanted to give John Dunn the opportunity to provide some welcoming remarks if you'd like. John?
I'm good, Steve.
All right.
I'm totally confident and thankful. Things are in your hands.
All right.
WFO.
All right, great. Well, thanks a lot. Well, good afternoon, everyone. Welcome to the science sessions. I'm Steve Rottenborn, and I'll be moderating the science sessions this year. I've been coordinating with all the presenters over the past few months, and I can say that we have a fantastic group of speakers lined up over the next 4 days. As you can see in the conference agenda online, the science sessions will be held for about an hour, starting at 12:30 and 3:30 each day of the conference, except that on Saturday, we'll have just the 12:30 session, as the WFO members meeting will be held at 3:30 that day. I also want to call your attention to the agenda tab on the conference website. The science program has been added at the bottom of the agenda, and the science program provides the schedule for science session presentations, abstracts for the presentations, contact information for the authors, and brief bios for the speakers. So please take a look at the science session or the science program when you have a chance. Each presentation will last about 20 minutes, including time for a few minutes for questions from the audience. So if you have a question for one of the presenters, please type your question using the chat function during the presentation. And at the end of the presentation, if we have time, I'll read some questions and the presenters can provide a brief reply. If you have questions and we don't have time to get them fully answered, please feel free to contact the presenter or one of the co-authors, as their email addresses are provided in the science program. So let's kick things off. And to start things off this year, our first speaker will be Ben Sonnenberg. Ben received a degree in biology from from Pacific Lutheran University in Tacoma, Washington, and he's currently a fourth-year PhD candidate at the University of Nevada, Reno, where he conducts research in the Avian Cognitive Ecology Lab, focusing on mountain chickadees. Ben is also, as John Harris indicated, one of the 2021 recipients of a WFO research grant. And today, Ben will speak to us about developmental stress and spatial cognition in wild food caching chickadees across elevations. So Ben, please start things off for us.
Hey. Everybody can see my screen okay?
Yes.
Okay. Well, thank you so much for that introduction. I really appreciate it. That's right, I'm from the University of Nevada, Reno, and we're going to talk about some mountain chickadees today. We're going to get started with a cognitive ecology talk, which is really fun. But first off, before we get started, I just want to say a really big thank you. Part of this research was absolutely funded by you guys, and namely the molecular component. And so this work was made possible by the WFO, and I really appreciate the support. A lot of my dissertation research is focused around how heterogeneous environments shape organisms during their early development. And specifically, I am interested in mountain chickadees and one particular phenotype, which is their spatial, cognitive, and memory abilities. And that relates to their food caching abilities, which I'll talk a little bit about in more detail. But this is, how heterogeneous environments shape complex behavioral traits is something that really has not been fleshed out in field studies. It's mainly been done in the lab. And so, for example, in rats and near model avian systems, this work has been done, but never in the field. And at least in the lab environment, there's a unifying theme. And there's a lot of research. And basically, the research says that in early development, if an organism experiences a developmental stressor of some sort, that impedes their future cognitive abilities. And so we really wanted to know whether or not we saw those same patterns that the lab studies reported, but in a wild system. That brings us to mountain chickadees. And so hopefully many of you are very familiar with these wonderful birds. These are residents to Western North America in montane coniferous forest habitats. And I happen to study a population in sage hen experimental forest, which is north of Truckee, California. And chickadees are an amazing species, and why we are so focused on this species is because of their food caching or food hoarding abilities. And so specifically, mountain chickadees are what are called scatter hoarders. And so they hide tens of thousands of individual food items all around their forest habitat. And so this is actually a series of photos showing a mountain chickadee making a cache in the bark of a mountain hemlock. But they make tens of thousands of these caches, and then they rely on advanced spatial learning and memory abilities to go back and retrieve those caches and consume those caches in order to survive harsh winter environments in the Sierra Nevada mountains. And so our particular field site is unique in that we study mountain chickadees, a population of mountain chickadees that resides across a montane elevational gradient in Sierra Nevada. And our field site is characterized by what's called a snow line. And a snow line is an approximate elevation for us, marked by that red dotted arrow, right, or line rather. It's an approximate elevation during transitional periods of the year, such as spring and fall months. Precipitation is much more likely to fall at high elevations above the line as snow, and much more likely to fall below the line as rain. And what that translates into is that higher elevation environments experience colder temperatures, more snow for longer periods of time, and it just translates into an overall harsher and less predictable environment than some lower elevation environments. And why this matters is because mountain chickadees are caching and trying to survive across this mountain elevation gradient. And from our work, we've shown that mountain chickadees are actually really highly resident. So birds that are living at these high elevation sites don't fly down the mountain to greener woods, if you will, in order to get away from the harsh conditions, but instead they live in these places and they deal with them year round. And so we can compare two subsets of the population at both a high and low elevation. And so when I say high versus low elevation for the rest of this talk, I'm referring to a subset of the population that's at 1900 meters and a subset that's at about 2400 meters. And so just to summarize some of the past work in this system, caching and specifically the spatial cognition, so the spatial learning required for cache retrieval, has long been hypothesized to be really critical for overwinter survival. And how do we go about studying this, right? And so first, we wanted to look at whether or not there was brain regions in the mountain chickadee that differed across this montane elevation gradient that might correspond to climatic harshness. And so brain regions, specifically the hippocampus. So the hippocampus is a brain region that's associated with advanced spatial cognitive abilities. And so we examined the brain morphology and see if birds with more space in their hippocampus designated to spatial learning had differences in caching, or if they had advanced, better learning and memory abilities. And we expected that birds at higher elevations might cache more and they might have better learning abilities because they have to deal with these more climatically harsh environments. And that's exactly what we found. The birds that had at high elevation had more and larger neurons in their hippocampus, and that corresponded to behaviors so they cached more, as well as they performed better on a spatial learning and memory task. And I'm going to talk about and break it down in detail how we actually measure spatial learning and memory abilities in a wild population of chickadees. But then critically so, we found several years ago and were able to publish that juvenile birds, so hatch-year birds in their very first year of life that perform better on our spatial learning and memory tasks were much more likely to survive than their counterparts, which is actually direct evidence that this trait, this complex cognitive behavioral trait, is under natural selection in chickadees. So this is a really critical and important behavioral trait for the natural history of this organism that allows them to survive in their habitat, especially during the winter. And that's why we're so obsessed with spatial memory. So how do we actually study spatial learning? Well, with really fancy bird feeders, of course. And so our chickadees are tagged with what are called passive integrated transponders. And so it's this white tag on this individual in the photo and the purple tag on the individual in the video. And so those PIT tags actually allow birds to interact with radio frequency identification equipped feeders. And so I paused the video so you can see, so this purple PIT tag is interacting with this black perch, which is actually an antenna that then triggers a mechanism inside a control board that allows this door to open and allow access to a food reward. And the chickadee grabs the seed and flies away. And so that allows us to actually test how well they remember. We can assign birds to specific feeders in an array of 8 that are arrayed in a spatial format, and allow us to test how well they might remember a specific feeder and its spatial location. And so what that actually looks like, so first off, birds, we tag as many birds as possible, and then we habituate them to our feeder system. So all birds with a PIT tag, all feeders open for those birds during the initial test. And that's just to get them habituated to the opening closing mechanism. But then as the experiment goes on, when it actually the test begins, we assign each specific bird that is visiting the feeders to one specific feeder in that array of feeders that they're visiting. And we break up flock mates to control for social learning. And then what the test actually looks like is, so when a bird comes to the array, after all the feeders have been previously rewarding him, he might visit a feeder that he no longer receives a reward. And he has to sample the subsequent feeders until he finds his particular rewarding feeder and where it's located in space. So there he's found it. He might take that seed, he might eat it, he might cache it, but then returning to the feeder, if he goes back to that same feeder, we know that he's learned, right, that spatial location. And so we can actually track the learning process. And what that looks like realistically so each time these birds visit the feeder array, we call it a trial. And so each trial is this, so this is a trial. And so they made 3 errors before they found their rewarding feeder. He made 2 errors before visiting, and then no more errors, right? And so if you imagine like a little curve right here, this is a learning curve, right? Improvement over time. And then we can test and measure by averaging all of these errors across, and some of these birds participate in hundreds of trials, so we can get a really accurate estimate of their spatial learning and memory ability. And so that's the metric that we use to measure spatial learning, which, remember, is really critical for these birds. And so if their cognition is being affected by early development, in which we do see lots of variation, it will make them less likely to survive in these environments. And so we're trying to understand where some of the variation that we see in learning abilities comes from, and whether or not that's from an early developmental environment. So actually, how do we detect differences in the developmental environment? And we go to this adorable, this is a little 16-day-old chickadee. And so we track these guys during their development, but we actually take a sample of cort. And so cort is shorthand for corticosterone. And so corticosterone in general, we're not going to get into details today, but in general, it's higher levels of this particular hormone is an indicator that this bird is experiencing higher levels of stress. And so more corticosterone, more stress in the early developmental environment. And a lot of people sample levels of cort by taking a sample of blood and looking at the levels of circulating hormone in the peripheral bloodstream during development. However, that only gives you one snapshot during the developmental period. And so we chose to actually sample feathers. So feathers are amazing in many, many ways, but feathers are amazing for this research in that, so as feathers are grown, the circulating levels of stress hormone are actually deposited and kept like a little time capsule of information in the keratin tissue of a developing feather. And so after these feathers are completely grown, we can pluck a tail feather and then, using a methanol-based extraction, measure the amount of stress hormone present. And that's essentially an average of the court experience, or the average of developmental stress across the entire development, especially early development of the bird. So it's a really fantastic method. And we measure that using a standard ELISA assay. So we predicted we would see lots of variation between and within elevations. And we also predicted that higher levels of court would correlate to poor performance on our memory task. And that's exactly what we saw in the laboratory literature, but we wanted, and so this prediction that's where that prediction is coming from. So it's a very straightforward prediction. But what did we actually find? So we did see lots of variation in corticosterone deposition. And so if you look at this graph, so this is 3 years of data, 2018 to 2020. This is corticosterone picograms per millimeter of feather. And the circles are high elevation and the triangles are low elevation. And you can see that there's lots of variation, but we actually didn't find any differences between elevations. So despite having climatic differences between these subsets of this population, they had on average, really similar corticosterone depositions. However, we did see differences between years, with 2020 having the lowest corticosterone deposition. And we still don't really know why we saw that pattern, and we're trying to tease that out. But what about the actual behavioral traits? What about cognitive performance? And so this is a new graph, right? So corticosterone is on the x-axis this time. We still have the 3 years of data, but location errors is on the y-axis. And so basically, the higher you are on the y-axis, the more errors the bird's made, so the poorer performing that they are. And if you look at this really steep, this line with this really steep slope, you can see that we actually did find an effect, but only for 1 year, and unfortunately due to sample size, only at one elevation. But it was still a very strong effect, and it was exactly as we predicted. So birds that had higher amounts of corticosterone in their feathers performed worse on the spatial learning and memory task. And so that's exactly what the lab environment found, and so that's really interesting. But what about the 2 other years? Well, we didn't find a pattern there. We had better sample size at low elevation in those 2 years, and we didn't see any pattern. And so what does this mean? So we saw it in 1 year, but not in the other 2. And so just in summary, right, we did see lots of variation, with 2020 having the lowest levels of court. And there were no differences in court deposition between elevations across the years, but we did see higher levels of court correlating with worse performance on a memory task as we predicted, but only in a single year, only in 2018. So what made 2018 unique? And how do we explain this variation, right? It's likely due to local climate. So the large variation in snow and temperatures across years is likely tied to year food availability, both the abundance and the diversity of invertebrate prey that chickadees are feeding their offspring. And we're just starting to measure that, and so we're not able to include that in this study. But just to show you an example of how invertebrate prey could be affected, so on the y-axis, so this is 2017 through 2020, and I included 2017 because 2017 had the largest snow levels in the last 100 years, and that was the winter leading into the breeding season of 2018 where we saw the effect. And so we talked to our invertebrate expert colleagues, and they said that they saw specific groups of insects that were hit really hard by these high snow levels. And so it could be that there was a specific prey item with a macronutrient that these chickadees target to feed to their offspring that caused the effect that we saw in 2018. And then it was a really light winter and possibly gave the insects the ability to recover their populations. And so then maybe the patterns, that's why we saw the patterns disappear. What we can't get a sense of that exactly what they're feeding their offspring, but we can get amounts. And so that's the other part of the study. And unfortunately, I don't have the data to show you right now. It's all still in the works. But it's a really exciting, we can still get more information out of these feathers. And it's through this method called telochronology. And so it's essentially like tree rings, except for bird feathers. And so if you look at these red lines here, you can see a dark bar and a light bar and a dark bar and a light bar. And those are growth bars of a feather that indicates a 24-hour growth period. And laboratory and experimental results have shown in the past that the width of those bars is directly related to how much insect, how much prey, right, what these individuals were eating while they were growing the feathers. So the wider the bar, the more that they were being fed by their parents, in this case of the chickadees. And so we still have to look at the telekinology, the feather growth rates, and whether that relates to stress levels and whether that relates to cognitive ability. And so that's some of the future directions. And we also, and it's just like a teaser, kind of a fun thing to mention, is we're specifically going to look for spiders in our population. And that's because spiders have high amounts of a molecule called taurine. And taurine in the laboratory environment has been shown to be what's called a neuroprotectant. And so essentially, animals that have high stress in the lab, that normally would experience a cognitive decline, have neuro rescue when they are given a taurine. So it acts as a neuroprotectant, and they develop the same cognitive abilities as if they were not stressed. And it's been shown in blue tits in the UK that blue tits preferentially seek out a certain amount of spiders to feed to their offspring. And so what we could be seeing in chickadees is that they might be specifically focusing on invertebrate groups to feed to their offspring to protect, because this is the spatial learning and memory abilities is so critical to their survival during the winter months. So just overall, we found that there was an association in developmental stress and spatial cognition. It just wasn't consistent across years and conditions. So surprise, surprise, nature is messier than the lab environment. But this gives us lots of future directions and areas to pursue. And we think that this lack of pattern might be just due to the fact that food caching species, due to the importance of the trait, have evolved a mechanism that protects the spatial cognition and the development of spatial cognition. And this might be having to do with diet composition involving some key nutrient that would be critical to that mechanism. Okay, so after all that, thank you very much for listening, and I will absolutely take any questions. And please feel free to email me as well if we don't have time, etc.
Thanks a lot, Ben. That was fantastic. There are so many different aspects of all that. I don't see any questions in the Q&A section, so I'll ask one. Just, you mentioned looking at spiders and taurine. What are some of the other future directions that you're thinking about with respect to this work? Mountain chickadees, or are there other species you'd like to look at this in?
Well, so some of the future work hopefully coming down the line would be comparing different populations. And so a collaborator at the University of Colorado Boulder, we're working with them to look at whether or not there is difference in cognitive abilities in blackcaps and mountain chickadees. Of course, that's out in the Rocky Mountains, and there's a hybrid zone between those two species. And so whether or not there's differences in cognition in the hybrids, whether or not there's differences just in the populations out there, in comparing those two species. And so that's a really exciting future direction that our lab is taking that leaves mountain chickadees a little bit.
Gotcha. It was also interesting how variable things were with respect to the court levels within a year and between years, and the patterns that you saw. Obviously very complicated and a lot more to learn, but this is fantastic. It's I've been seeing your work for a couple of years and just been fascinated by it.
Well, we appreciate it. That's awesome. We're really excited. Obviously, it's I'm one of many lab mates in the group, and so there are lots of different directions. We're looking at social networks. We're looking at social learning, right, and how all of those things relate to cognition. And so it's a really big study, and we're just like answering more questions every day. And I just want to stress to everybody that mountain chickadees are, greater than they appear.
They absolutely are. All right. Well, Ben, thank you very much. Again, to the audience, just a reminder, please feel free to type in questions using the Q&A. Oh, I see. Sorry. I was looking at the chat function. So, all right. So here are a couple questions for Ben. One is, could climate change alter how the chickadees live?
Yeah, so that's a really interesting question. And so we can't answer that yet, but we've been measuring the the learning abilities and the caching behavior of chickadees now for over 7 years, and we're going to continue to do so. And we have several SNOTEL stations that are taking climate data that are directly within the field site. We have yet to see any specific changes in behavior, but we're hopefully going to be able to answer some of those questions in the future. It of course would change if the high elevation environments would just become much nicer to live in. It would very much decrease the selective pressures on these chickadees to have such accurate spatial memory abilities anyway, but we have yet to see that.
All right. Another question, do chickadees cache only seeds or do they cache invertebrates?
Yeah, that's a good question. So chickadees really are only caching seeds. So they cache really, really heavily in the fall and early winter months. And so all the invertebrates, they switch over to being primarily insectivorous, mainly in the summertime. And so they're primarily granivorous in the winter.
All right. And another question, would the 2018 correlation between stress and cognitive ability persist if a few outlier individuals, for example, at the upper right, were excluded?
So that's a good question. So all of that data, those are statistically not outliers, but that's a good question. So if we do remove those, we still get a really strong effect in that 1 year. We can't get rid of that effect in the 1 year. So yes.
Gotcha. And then finally, John Dunn indicated some people believe that the mountain chickadees in the Rockies are a separate species.
John, what do you believe? That's what I want to ask. So I had a pleasure of hanging out with the young birders and speaking to John about some of these ideas earlier this year, which was fantastic. And I don't have strong opinions of whether or not they should be split at this time. So I can't speak to that.
Gotcha. All right, Ben, thank you so much. That was fantastic.
Thank you.
All right. So our next speaker is AYALA Berger. AYALA was a 2020 recipient of a WFO research grant, and she's a PhD candidate at the University of California, Riverside. AYALA studies the evolution of complex courtship displays in Anna's and Costa's hummingbirds, and today she'll be speaking about geographic variation in Anna's hummingbird song. So welcome, AYALA.
Thank you. Let me just, sorry. 1 year in, you'd think I would know how to do this already. Thank you for the introduction. I'm really excited to be here. A whole weekend of birds is very exciting. So today I'll be talking about geographic variation in Anna's hummingbirds. So animals rely on sound for communication. The sender must emit a signal that modifies a receiver's behavior, and the signal has to travel through an environment. So animals rely on sound to find mates, seek food, establish territories, and rear young. And we know that there's geographic variation in a lot of acoustic communication. So based on Marler and Tamura's classic white-crowned sparrow work, Nelson et al. Expanded the study and found dialects in white-crowned sparrows. There's also evidence of geographic variation in marine mammals, such as the South Pacific humpback whale. And we as humans also experience geographic variation, whether we say store or sta, third or thoid, or wash or wash. We interact with geographic variation in how we speak a lot. So for variation in vocal parameters to be considered dialects, we expect within across geographic localities for there to be these sharp borders in these vocal parameters rather than a clinal variation or random variation. There are three main hypotheses for how and why dialects develop. So the social adaptation hypothesis hypothesizes that males gain fitness advantages by singing songs similar to those of other males in the region. And that males who are not singing the local song might be subject to penalty. So under this hypothesis, we expect individuals to learn vocalizations upon dispersal. So once they come to the new area. The epiphenomenon hypothesis with no adaptive significance essentially says that dialects develop due to cultural drift or random change. So there's no adaptive significance to having dialects. And the local adaptation hypothesis hypothesizes that females gain fitness advantages when they are able to mate with males from their natal regions. So essentially songs might be showing as a signal of nativity to an area. So dialect research has been biased towards passerines. So even though there are three distinct lineages of song learning in the passerines, the Pistacidae and Trochilidae, much of the research has been biased towards these passerines, and not much has been studied in these non-passerines. So today I'll be focusing on a species in Trochilidae, the Anna's hummingbird. So Anna's hummingbirds have a range that covers much of the Western United States. They have a hypothesized lek mating system with uniparental care. So there's no paternal care involved. They also have multimodal courtship displays, incorporating visual and acoustic displays to each other. Their song is complex, three-phrase, and multisyllabic. So Anna's hummingbirds have a recent range expansion from the 90s to 2013. So within the past 30 years, the range has expanded northwards pretty dramatically. And this is due to supplementary feeding, which has allowed Anna's hummingbirds to occupy habitats in the winter that might have previously been unsuitable. So here we have an Anna's hummingbird sitting on a feeder with snow on it. And this coincides with the breeding and nesting season. So the breeding season for Anna's hummingbirds begins in the winter. Anna's song is very complex. So there's these three phrases, phrase A, B, and C, with multiple syllables within them. So I like to play a sample of what the song sounds like, and I'll follow with my error where we are in the spectrogram as the song goes. So to really appreciate how complex this song is, if you don't appreciate it visually, I will also like to play this same clip slowed down to half speed. Really complex songs. It makes the passerine songs look simple. And so previous research has looked at microgeographic variation within Anna's hummingbirds. So Yang et al. Studied a population of birds in in San Francisco. And they found a lot of variation within phrase A and not much variation within phrase C. And so this is a population at Golden Gate Park, which I've resampled 20 years later. So this brings me to my research question. And the question is, do Anna's hummingbirds have dialects? To study this, I recorded 6 to 24 birds in 6 different populations with 10 to 30 song iterations for each individual. And I chose a spread of populations from Seattle, Washington in the north, which represents a recent range expansion, Mendocino, California, San Francisco, Santa Ynez, Riverside, California, and Henderson, Nevada. And I used a parabolic microphone to record these birds. So I've conducted preliminary spectral analysis where I focused on 3 elements of the song. This is B1, B3, and C1. So for B1, I found that here in Riverside, we see this presence of B1 here, whereas in Seattle, we do not. And so what I found was that B1 is present in all populations but Seattle. But in Mendocino, B1 presence is variable. That is that some individuals sing B1, whereas other individuals do not. And it's interesting to reflect back on the map where Mendocino is an intermediate population between San Francisco and all the other populations and Seattle. So it seems like there might be a longitudinal trend. For B1, in the populations that did sing it, I looked at peak frequency and found no significant effect of population on peak frequency. Moving to element B3, which I measured the fundamental frequency, and it consists of these pulses. I looked at first the frequency, the peak frequency, and found a suggestive effect of population on frequency. And then I counted the number of pulses and found a strong effect of population on pulses. And it seems to be that there's a longitudinal trend here as well. And then I looked at pulse rate and found that there is no effect of population on pulse rate. I then looked at the next element, C1, which are these 2 pure tones, and I focused on the fundamental of the first pure tone and found that with both peak frequency and duration, there is no significant effect of population on peak frequency of C1. So conclusions to date, I looked at presence and absence of this first element, and this indicates a strong effect of population on whether this whole element is song, which might suggest sharp borders between populations. And it strongly suggests that there are dialects between different populations. When I looked at the peak frequency of this fundamental frequency, I found no significance. So it's interesting to look that there are both areas where the song seems to be significantly different between different populations, but also seems to remain the same. So there's variable elements and very similar elements that stay the same. So far, we have evidence of dialects on a macro geographic scale. And here are some photos from 3 different field sites. On the top is Seattle, Washington, and it actually snowed while I was recording there. In the middle is Riverside, California, which is the chaparral coastal scrubland habitat. And then on the bottom is Henderson, Nevada, which is dominated by this mesquite. So it's interesting to look at these 3 really distinct habitats and think of the songs in relation to them. There's evidence for a longitudinal pattern in some elements, but not others. So there's evidence of a longitudinal pattern in B1 and B3. And this represents a new addition to what we know about dialects in non-passering. So essentially we can add Anna's hummingbirds to this class that has geographic variation. So if I haven't convinced you that Anna's hummingbirds are really interesting in their songs yet, we can broaden this discussion to thinking about their behavior. So Anna's hummingbirds lack paternal care, so understanding who they're learning their songs from is quite interesting. They have a hypothesized lek meeting system, although it's likely an exploded lek. And so understanding how this system influences dialect development is interesting. And they represent a recent range expansion. So how this affects dialect development and whether you can gain knowledge about their dispersal patterns based on the songs they sing. What I've left off is another part of the story, which is that Anna's hummingbirds both sing and do these dive displays in which they fly up and then dive towards a female. And while they dive towards the female, they make a sound with their tail feathers. And this sound is produced with their tail feathers and their wings. And so here on the left, we have their dive sound of Anna's hummingbird male. And we see that there's multiple elements. There's really 1, 2, 3, 4 distinct song elements. And it seems to match with the song that the Annas are singing. So this is the same individual singing a dive-like portion of the song, and we see similar elements here. And they also correspond to 2 elements that I measured. So I took a very conservative approach to preliminarily looking at my recordings by measuring 2 elements that was in this apparent match. So my future directions are to measure more elements of the song and to include dive sounds in my measurements, and to look at variation in dive-like sounds and whether there's similar geographic trends. I'm interested in studying how these dialects or likely dialects interact with a match in signal form. And with that, I have many people to thank. Of course, the hummingbirds, Western field ornithologist for the grant money that really allowed me to sample all the populations on the Western hemisphere or Western coast. So thank you.
Thanks a lot, Ayala. I will readily admit that I was ignorant of just how complex their songs are. I've got to ask before we get to some of the other questions, just because until you slowed the song down, just how diverse those phrases are, I couldn't hear it well. How did you come up with the idea of studying this in Anna's hummingbirds of all species?
Yeah, so I'm studying this actually both in Anna's hummingbirds and Costa's hummingbirds. And so Anna's hummingbirds really drew me because they are so complex. And they are like proven song learners. We expect there to be variation. And when you think of the story broadened out to also including a sister text of the Costa's hummingbird, which has a very simple 1-phrase song, and also are song learners, it's interesting to look at the 2 simultaneously. So.
Gotcha. All right, thanks. Some of the Q&A, John Dunn says, I'm not aware that the congener Costa's hummingbird has no song unless it is the thin rising and falling sound, yet they rather frequently hybridize.
Yeah, so Costa's both dive and sing, and the match is much stronger in Costa's auditorily. And interestingly enough, hybrids of both Anna's and Costa's usually tend to sound like an Anna's hummingbird. And the dive display that they do is sort of intermediate between the 2.
Interesting. Well, along the same lines, Kimball Garrett asks, might mimicry contribute to uniqueness of some dialects? In his area at the north base of the San Gabriel Mountains north of Los Angeles, male Annas all seem to incorporate quick pit-pit notes in phrase A, which sound identical to call notes of costas. So could mimicry play a role?
That's really interesting. Yeah. I assume that mimicry plays a role within the song learning aspect. So it's really unknown who they're learning their songs from. So yeah, that would be really interesting to work and look at.
And then there are two similar questions. Are there other hummingbirds that have geographical differences in their songs?
Yeah, so there are other hummingbirds not in this clade of hummingbirds, not in the B clade of hummingbirds, but there's the black Jacobins who have found to have geographic variation in song, and also little hermits who have geographic variation in song. And they have like really strong evidence of like variation in between neighboring leks.
And then Kimball Garrett also pointed to a publication on a unique Anna's hummingbird dialect on Cedros Island off Baja. He said it might be worth looking at other island populations.
Yeah, thank you for that suggestion. That paper is so cool. Unfortunately, the island is nearly impossible to get to. But yeah, they had a strong evidence of dialect and did a playback experiment. So future research would also incorporate some sort of playback to see if there's a behavioral response to variation in the songs.
Well, if you're talking about island populations and hermits, maybe it's time for another WFO research grant. Yeah.
That would be the ideal.
All right. Ayala, thanks a lot. We really appreciate it. Fantastic talk. So those were both outstanding presentations. So thanks to both Ayala and Ben. John, do you want to say something about what's up next?
Yep, so we'll be taking a short break and we'll reconvene the conference at 2 o'clock. Our next speaker will be Helen Laughlin speaking about birds and meadow restoration in the Sierra Nevada. So we'll see you hopefully at 2 p.m. Thanks very much, Steve.
Thank you all.